Okay, so where are we? We introduced formalism based on the language of chemical reaction in order to describe cellular processes, molecular processes, genetic processes. These formalism can be used to build models of what we attempt to describe. There are actually many ways to model, many ways to formalize it, but each time it is the building of a model in a given language, here the language of this chemical reaction or pseudochemical reaction, as each reaction is not a pure elementary reaction between molecules, but can be reaction between huge objects. It represents a process. Each reaction represents some step in the biomolecular cellular process. Now, this formalism enables to describe with more precision a diagram of the or knowledge, a drawing of the knowledge, and then enables to translate each step that was described into a mathematical expression. So now it's this last step of translation in mathematics that we will do together on the very easy first example we already saw of the activator, regulator, transcription factor. So the question, back to our activator diagram, is how the concentration of mRNA varies in time given some concentration of the activator transcription factor. Okay, now on this slide we see again the translation in terms of chemical reaction of the diagram above for an activator A transcription factor and production of mRNA only when the promoter is occupied. Here the last reaction is we choose to include here also degradation of mRNA. Choice to include this into the model. Applying chemical kinetics, we can easily write a differential equation to describe the change of the concentration of messenger RNA in time, depending on, well, the reaction where the messenger RNA is produced. Here, the second reaction. So, seeing this as a first order reaction, the differential equation for messenger RNA concentration is that it is expressed at some given rate, given by some kinetic parameter, K, and okay, since here there is another reaction where messenger RNA is involved, the degradation, in the differential equation there is the term for degradation, which is here this order, second order equation, first order equation, sorry. So simply applying chemical kinetics, we can generate a differential equation for the evolution in time of our gene product, here the messenger RNA. Okay, but our initial question was how is a messenger RNA produced as a function of the available transcription factor A. So what is still needed to answer the question is to compute what is this concentration of bound transcription factor as a function of A. So this is still to be solved and has to be solved looking at the reactions that produce the bound promoter. We have to write the whole differential equation system. Make a pause in the video and try to translate all these chemical equations into the differential equation for all the species involved. Try to do it yourself. Okay, let's try to do it together now. We must now translate the promoter binding reaction we have here 3 species, so there will be the variation of concentration of the 3 species involved: activator, the promoter, and associated promoter. And the reaction is forward reaction is a second order reaction, so it will involve some parameter K plus times the product of the reactant. It is here a reaction that uses A, so it will be negative for the concentration of A, and each time one A is used, 1 mole of A is used, there is a production of a bound promoter. So we find the same expression here for the rate of destruction of A for the production of bound promoter. Then for DNA it will be the same, both react in the same way. And since we have dissociation, there is the reverse reaction, which is a first order reaction. First, we will see it here as a degradation of a DNA, or the bound promoter, negative for this equation variation of bound promoter concentration, but will be contribute positively for A and free DNA. Okay, this is standard, we did it already. Now there is still the reaction for mRNA production. Okay, and in this reaction, what do we have that changes? Messenger bound promoter does not change, so there will not be no contribution from this reaction in the equation giving the variation of bound promoter. But there is the additional new species, messenger RNA, that is produced. That is actually what we would like to have for a result, an equation for messenger RNA variation in time. It is a first order reaction with some other synthesis parameter. I will call it K2 plus, or simply K2 to distinguish it from the other parameter there. And this is, well, it was already on the slide, proportional to the concentration of bound promoter. And messenger RNA can also be degraded. It's the last equation, the last reaction we choose to include in our model. This is a choice, what you include in the model to describe your process. And so we have here to include the degradation term, which is first order reaction proportional to the amount of messenger RNA. We saw this last equation already. So finally, here is the whole system of differential equation that describes our simple model of an activator. And the issue is that, okay, we have here 4 differential equations to describe at the end the quantity of interest would be this: how does messenger RNA evolve in time? So it's not explicit from this system how will messenger RNA concentration evolve in time given some concentration of transcription factor. For even this simple model, you see the amount of equations that are involved. It can be solved numerically if we can find values of all those parameters. And one of the big tasks of modeling will be to find values, biological meaningful values for the different parameters. But even then, okay, it will be not very straightforward to see the result of interest. In the next video, we will see how, starting from this 4 differential equation system, we can come to a simpler expression, so a simpler system that will be more efficient, that will capture some of the dynamic in a more simplified